So, good morning, one and all. Respected Chair, Dr. Sesikaran, colleagues, friends. Like Dr. Sesikaran said, cotton was the first crop, and a lot of experiences to share. I have my colleague Bharat and my colleague, Sanjay Deshpande, who has really more stories based out in Delhi about BT cotton and experience. Just to start with, I know this might look like a bit old, but I thought let us, reinvent the wheel and go back to what was the rationale behind working on BT cotton. We all know, we had this is the pre-BT cotton era, story, just to get you in the room. Estimated 4 million cotton farmer, 60 million people depending on, industry, textile industry. We are the number one, in fact, in terms of the textile revenue. We plant more cotton even today, and we used to plant it more than anyone else in the world. Though we had 25% of the world's cotton, we only produce 16% of it. Our average yields were around 300 kilogram when the world was producing about 600 plus. Losses more than 30% due to various insect pests, disease and weeds. The figure when we launched in 2002, just prior to that, it was estimated that approximately 13% of cotton was lost. In terms of volume, it was more than 15 or 16 lakh bales. And then in terms of value, more than 1,300 crore rupees only for one season. And it was estimated that 12 billion worth of pesticide was used in a single year to control the bollworm complex. This was the estimated one. A very interesting figure, friends. Total pesticide consumption 83,000 tons. On cotton it was 42,000, and alone on bollworms it was 31 or approximately 32,000 tons. What could have been the estimated savings post use of BT cotton? Tremendous. I'll just give a number. The estimated, and today it was thought of 25,000 tons would have been saved. But today I tell you it is more than 39,000 tons which have been saved post the launch of BT cotton. A journey which started in 2002, the first ever transgenic crop which was commercialized in India. If I remember the date, it was 26th March 2002 when we got the approval to plant the first BT cotton commercially. First year 0.72 lakh hectares and lakh acres, and today, last year, we were 270 lakh acres. In terms of million hectares, it is approximately about 11 million hectares of cotton under BT cotton today, which is close to 95%. Just an example of one state, Maharashtra, which plants the largest cotton anywhere in the country. It is approximately 100 lakh acres now. So that can be the impact of any technology in a span of 10 years. We have, the acreage is more than 95%. How things have changed. Like I told you, 2002, our country's production was about 140 lakh bales. Today, last year, we produced 334, and I believe this year, because of the good rainfall, we expect more. Our productivities have increased from 300 odd to approximately 500 kilogram per hectare. Acreage, as I told you, close to 11 million hectare, and we were a net importer of cotton in 2002, and now we export cotton to the tune of 80 lakh bales, in which we have, we are now the second ranked country in terms of cotton production in the world. Earlier we used to be fourth, we came up, and we are now the second. Just an example of a state, how things can change again. If you see the acreage, pre-BT cotton era in Maharashtra and the acreage now, the acreage itself has increased by approximately 11 lakh hectares. The productivity has increased. It's like from 195 kilogram per hectare to 353 odd kilogram per hectare. And the production from 34 lakh bales in Maharashtra, it produces now 85 lakh bales. What were the guidelines which were followed when we were working on, BT cotton? The first two, the recombinant DNA guidelines for 1990 and 1998, mainly were used for BT cotton when we were working on cotton. Today, we have the 2008 guidelines, very robust guidelines which have been prepared. It all started in the year 1995. When we got the permit to import seed from US, and in 1996 and 1997, I was going back to the data, and 3 years pollen flow trial data was conducted, and it was not a single year to reconfirm that to how much extent the pollen might flow from the BT cotton plant to the non-BT or to the neighboring areas. So three years data, one in 95, one in 97, and then again in 2001 as per the new protocol. And in the best part was at 97, it was planted at 4 locations to see, is there any locational change from central India to South India to North India. So that was evaluated. Friends, I'm sharing with you the whole story about how it happened. So it might sound, a bit lengthy, but this is the fact, what had happened. We came in 1998 and 15 trials of multi-location trials were permitted in various states. The interesting parts are the states which I have mentioned in the parenthesis. And then in the same year, we were also allowed to plant large-scale trials, large-scale multi-locations at 25 locations, again across the country. We submitted the data in 1998 and 1999, a new terminology of multi-centric trials. They were called as multi-centric trials. So at 11 different locations across 7 states were, permitted, and then we started the biosafety studies. The first study which was done was a 90-day goat feeding study which was done at ITRC Lucknow. The protocols were approved by RCGM, ITRC, and then the study was done in 1999. It was on goat. 2000 again we were permitted trials. There were ICR trials and multi-location trials which were done by Mahyco. 14 multi-location trials, and simultaneously it was informed that we do large-scale trials. So it was like you go to the farmer field and this trial should be non-replicated. Now we want to see what could be the impact of a half-acre field trial actual on the farmer's life after he plants BT cotton. And across 5 states we were permitted to plant these trials. In 2001, we submitted the dossier after doing all of the biosafety studies in 2000 and 2001. 2001, the studies which we did, so it was informed to us that the studies are enough, but we need to do a really robust and large scale. 376 trials were planted in 2001 across India, right from Punjab to Tamil Nadu. And research, again 1 year ICR data and university trials were planted at 14 locations in South and Central Zone. Simultaneously, from the 2000 data which we collected, we did the molecular characterization on the first 3 hybrids. These were the first 3 hybrids in which BT cotton was approved in India. The confirmation of the absence of the terminator gene, that was considered very important in those days, and we were supposed to do that because that was a lot of hue and cry that it carries terminator gene into it. Then what could be the feed is consumed by the cows and buffalo. So it was decided that it was not a mandate, but what could be the impact on the lactating cow and buffalo. So suddenly it was mentioned to us that you need to do a study on lactating cow and buffalo. So we did this study at NDRI Karnal and Pantnagar for buffaloes. Then the effect of cotton seed produced again, which was done at NDRI. Then it was like, what would happen, again on the feed. So one more animal model. I'm just trying to brief you, and Dr. Sesikaran will agree. So many different animal models were evaluated. Then chicken came into picture. That it was suggested that let us do a study on chicken. So we went to Kari and we did a study at Kari Izzat Nagar. The most interesting study, it was like, what could be the impact if the BT protein was fed to the fish? So imagine, fish eating cotton or the cotton protein. So a study was done at Sifi in Mumbai, wherein it was said that you need to get the BT cotton seed crushed and it should be used in the catfish. So we did that study. Then the stability we started the trials in 97, 98, all the field trials until 2001. So stability of the gene, Cryvan-AC gene. Again, a pollen flow study, like I said, different diagram of how would the study be all together, making blocks around the BT cotton plot and evaluating it. Overall, we did the expression levels were evaluated. Then it was a very important point came up that what could be the socio-economic impact once BT cotton is commercialized. Again, so at the last moment we were informed that no, we need to do a farmer survey of all the trials which we had till date, and we need to evaluate a socio-economic study. And then it was done through IIM Ahmedabad. In addition to this, various aggressiveness, weediness, germination, soil microflora, NTOs, beneficial, the studies were done from 1997 to 2001. Some of the pure protein studies, which we got because the gene came from Monsanto, so we got the pure protein studies, the digestibility in simulated gastric fluid, acute oral in albino mice, parasitic Hymenoptera, honeybees, ladybird beetle, and lacewing larvae. These were all the pure protein studies we got from Monsanto. And in addition to that, in India, again, allergenicity in guinea pig, brown Norway rat, and Sprague Dawley rat, a 90-day study was done. All put together, approximately 22 plus studies were done to evaluate and prove that BT cotton is as safe as non-BT cotton in India. Friends, I was going through the technical agenda, and I thought this slide could be one, I felt could be one summarizing slide of how an ideal risk assessment which is being done today in India can be and should be taken forward, which we are following even today. So I have divided it into three parts: the laboratory greenhouse studies, or the confined field trials, and then the environmental release. So what do we mean in the initial phase? Ideally, the acute oral, thermal, pepsin, and molecular characterization is being done today. As soon as the material moves from the greenhouse or the lab to the confined field trials, so we start the various studies which are aggressiveness, weediness, impact on NTOs, impact on pollinators, soil microflora, and then in terms of the food feed safety study, we do a compositional analysis, a subchronic livestock feeding study, a subchronic feeding study, and a livestock feeding study, which, as mentioned in the 2008 guidelines, is a case-by-case study. Overall, what happens is these studies are done while the BRL1 trials, which is as per the new system, they are conducted. The BRL1 is conducted for 2 years, the first year and the second year, or 2 seasons, mind me. So these studies are evaluated and completed while the BRL1 trials are going on. Once the BRL1 trials are completed and the biosafety studies are done, the biosafety package is submitted to the RCGM. And then after the RCGM reviews it, it then goes to the GAC for the permit of BRL2 trials. And then during the BRL2 trials, we get the field performance and the relevant data which is asked. And then is the environmental release. So I believe this is the ideal which is happening today in India. This is, wonderful system which we have put in place now. I was just comparing, what happened in BT cotton or Bollgard 2 biosafety studies then and now, and also Bharat will cover in his, BT brinjal. So on the right side, if you see, the column is which, as per the old guidelines, we were asked to do so many studies. And as per the new guidelines, now in 2008, we have a mandate of what is required. And there is a yardstick to that you need to do in terms of the pure protein studies. You need to do a acute oral study, pepsin thermal stability or pepsin digestibility, that has to be done. And then on the plant protein, a subchronic, livestock, and compositional analysis has to be done. On the environmental part, it's the same, the soil microflora, germination, aggressiveness, weediness. But yes, there is a system in place now, and we feel it is robust enough. It can, it can match with any other best system in the world. So this was just a comparison of how it was earlier and how it is today. Need for field testing, as per the, I thought these two slides could share that, most of the risk assessment studies which are done under control, they are done, but however, this cannot substitute the field release or the field studies or the environmental studies, because you cannot 100% simulate the variability. Plant breeders, we all know, have been, testing their materials using the GBIE concept, and that's one of the most important thing when you release any hybrid or any variety. So for both efficacy as well as environmental safety studies, we need to have field testing, which is very important, and as you gradually go. So it is field to account for GBIE and real performance. Field studies are must. The key and as per the guidelines of 2008, it is very well mentioned that whatever the compositional analysis study has to be done, that has to be done from the material which comes from the representative locations of the field trials. So how can we just say that all the studies can be done in the greenhouse? The field studies are must, they are required, and these are the reasons why they are required. Just to take you to the future, I just thought I could share one slide with you. This is the upcoming technology on which we are working. This is herbicide tolerance and insect resistance technology, which we would be calling it as Bollgard 2 Roundup Ready Flex. And I thought our session today also has, we will be talking about stack events. So I thought this could ignite the discussion later on for the stack events. The center picture is the trial which we had planted prior to the spray of Roundup or the herbicide. You can see how was it. On the upper right side, it's the 4 days after spray, followed by 7, 15, and 21. Absolutely no interculture done on this field. It's only one Roundup spray, one herbicide spray. So this is the future for the cotton farmers. This is one interesting slide. I just thought ILSI has given us the opportunity of talking about BT cotton and BT brinjal. So I thought I would like to show that why it has been, or this is the reason, friends. Mahyco till date in India has conducted close to 3,777 various trials across crops, across traits. And I don't think anyone in India has, that rich experience of conducting trials, and like Dr. Sesikaran said, the nightmarish experience also. So this all came after conducting 3,777 trials. Just in brief, if only for people who are not aware about it, we would be entering the 50th year on the 11th November 2013. We are a 49-year-old company now. Our founder, Dr. Barwale, has been the World Food Prize laureate and a Padma Bhushan awardee. We have more than 200 products in the market. And we are the first in and the first in biotech crops also. And these are various products in which we are present across all field crops, vegetables, and cereals. And this is a happy cotton farmer who is planting BT cotton since 2002. Here I come to my presentation, and if there are any, with the permission of the chair, if there are questions after the session or right now. And I thank you, Dr. Sesikaran and Mrs. Rekha Sinha, for giving us this opportunity to present a case study and what we have done for BT cotton and how farmers' lives have changed after BT cotton. Thank you very much.